Welcome to the Development of Drosophila module. In this talk, we're going to learn about segmentation. Drosophila is a protostome that belongs to the phylum Arthropoda. It comes under the class of Insecta, and it should be remembered that only insects come under Insecta and not spiders and scorpions or other eight-legged creatures. These eight-legged invertebrates usually come under the class of Arachnida. In the case of insects, including Drosophila, the adult has a segmented body. In the context of Drosophila, when we look at the different parts of the body, there is the head followed by the thorax that is made of 3 segments. This is followed by the abdomen, which is also segmented, and finally at the posterior end the tail is present. This segmentation of the body that is observed in the adult is also observed during embryonic development, and thus embryonic development results in segmented larvae. The process of segmentation begins during gastrulation. During gastrulation, which are the early stages of development, parasegments are formed in the developing embryo, but these parasegments are transient. There are 14 parasegments that are formed, and these parasegments can give rise to segments. The way the segments are formed are the posterior part of one parasegment and anterior part of the next parasegment will fuse to form a segment. And that is what is shown in the blue box in the figure, where we observe segments being generated from the parasegments. The segmented embryo undergoes further development to give rise to an adult that also has a segmented body. During early development of Drosophila, the fate of the cells depends on protein gradients that are present in the syncytial blastoderm, and thus specification is flexible. However, as development proceeds and cellularization occurs, the cells start undergoing irreversible determination. The segmentation genes mediate the transition from specification to determination. When we look at what genes play an important role in the segmentation process, we have been able to identify 3 groups of segmentation genes. They include the gap genes, the pair rule genes, and the segment polarity genes. These 3 genes aid in the process of segmentation in the developing embryo. With this, we come to the end of our talk, where we learned about segmentation in Drosophila and how segments are formed from parasegments and the groups of genes that are involved in segmentation.